welcome to Biology, the scientific study of life, vegetation and nutrient cycling, the Hubbard-Brook experimental forest. In one of the long-running examples of long-term ecological research, LTER, in North America, a team of scientists led by Herbert Borman and Jean Likens has been studying nutrient cycling in a forest ecosystem since 1963. The study site, the Hubbard-Brook experimental forest in the White Mountains of New Hampshire, is a deciduous forest with several valleys, each drained by a small creek that is a tributary of Hubbard Brook. Bedrock impenetrable impenetrable to water is close to the surface of the soil, and each valley constitutes a watershed that can drain only through its creek. The research team first determined the mineral budget for each of 6 valleys by measuring the input and outflow of several key nutrients. They collected rainfall at several sites to measure the amount of water and dissolved minerals added to the ecosystem. To monitor the loss of water and minerals, they constructed a small concrete dam with a V-shaped spillway across the creek at the bottom of each valley, as shown in this figure. About 60% of the water added to the ecosystem as rainfall and snow exists through the steam, and thus the remaining 40% is lost by transpiration from plants and evaporation from other organisms and the soil. Preliminary studies confirmed that internal cycling within a terrestrial ecosystem conserve more of the mineral nutrients for example, only about 0.3% more calcium, Ca2 to the power 2 to the power positive, left a valley via its creek than was added by rainwater, and this small net loss was probably replaced by chemical decomposition of the bedrock. During most years, the forest actually registered small net gains of a few mineral nutrients, including nitrogen. In one experiment, the trees in one valley were cut down, and then the valley was sprayed with herbicides for 3 years to prevent regrowth of plants. All the original plant material was left in place to decompose. The inflow and outflow of water and minerals in this experimental altered watershed were compared with the inflow and outflow in a control watershed. Over the 3 years, water runoff from the altered watershed increased by 30 to 40%, apparently because there were no plants to absorb and transpire water from the soil. Losses of minerals from the altered watershed were huge. The concentration of Ca to the power 2 positive in the creek increased fourfold, for example, and the concentration of K positive increased by a factor of 15. Most remarkable was the loss of nitrate, which increased in concentration in the creek 60-fold, as shown this figure. Not only was this vital mineral nutrient drained from the ecosystem, but nitrate in the creek reached a level considered unsafe for drinking water. This study demonstrated that the amount of nutrients leaving an intact forest ecosystem is controlled mainly by the plants. These effects of removing the trees are almost immediate, occurring within a few months and continue as long as living plants are absent. The 40 years of data from Hubbard Brook reveal some other trends. For instance, since the 1950s, acid rain and snow have dissolved most of the Ca to the power 2 positive from the forest soil, and the streams have carried it away. By the 1990s, the forest biomass at Hubbard Brook had stopped growing, apparently because of a lake of Ca to the power 2 positive to set the possibility of growth limitation by inadequate supplies of calcium. Ecologists at Hubbard Brook began a massive experiments in 1998. They first established a control and an experimental watershed, which they monitored over 2 years before using helicopter to added CO to the Ca to the power 2 positive to the experimental watershed. As Jean Likens points out, the trees growing in Ca to the power 2 positive enriched soil are already showing signs of increased growth. Results of the Hubbard Brook studies, as well as 25 other LTER projects founded by the National Science Foundation, not only assess natural ecosystem dynamics but also provide important insight into the mechanisms by which human activities affect these processes.